Good afternoon, everybody. Hey, gang. Hey, St. Francis. Go, St. Francis. Oh, there's one thing I wanted to mention from this morning that I forgot to mention about our previous speaker, Greg Dunn, and that photograph that he made, self-reflected, or art piece that he made, that is on permanent exhibit at the Franklin Institute in Philadelphia. And if you haven't been there, that's like a really cool place. It's a science museum, and they have a heart that you can actually, I say heart where I'm from in Vermont, but they have a heart that you can actually walk through, and kids like to go to it and everything. But they have, it's a wonderful place. So if you've never been to the Franklin Institute, you should go there. And so I'd like to introduce our afternoon speaker, and Dr. Dell also received her Ph.D. from the University of Pennsylvania. I can wait. I got my teacher voice thing here going. Thank you. And she has initiated a project at the college called Art in the Lab, and along with Irina Ellison, who is from Mercy College. And she will tell you about this, and Mercy, and she will tell you about the project and about how you are all invited to come and participate if you'd like. And she's been teaching at St. Francis for the past 5 years, and she teaches neurobiology, and she has taught anatomy and physiology, general biology, and molecular biology. So she's has quite a breadth of knowledge. So here she is.
Hello. Good afternoon. Thank you, MacCube. Thank you to the organizers for inviting me to speak to you today. Thank you to the students who have worked really hard to put together posters and presentations. I was really impressed by some of the work that I've seen today, and I congratulate you guys. Taking the time and the effort to look at the work that you've produced, the scientific work that you've produced, and present it to your community is one of the best ways to learn science, because you're taking that science, that work that you did, the work that maybe relates to your coursework, and you are making it yours. So when I was thinking about the title for this talk and knowing that I was going to follow Greg's amazing talk today, I decided to think about the people I was speaking to, which is the teaching scientists and the students who make up the heart of MacCube, and to say, I almost titled this talk, Making It Yours, because I would like to talk about using art and science together as a way to make it yours. So I'm an artist and a scientist. I got my doctorate at UPenn looking at the cell and molecular biology of neurons, and particularly I looked at G-protein coupled receptor signaling in neurons. And now when I teach science, I have to think about how can I make it mine, and how can I help the students make what they're learning theirs. So I'm going to tell you a few stories today. First, I'm going to talk about a project that I started called Art in the Lab, together with my friend and collaborator, Irina Ellison. I'm going to talk about how scientists can use methods or art methods in their work, and talk a little bit about how artists use science methods in their work. And then I'll talk about a few art projects that I've done separate or in relation to these. Okay, so I'm going to start by talking about art in the lab. And here I really need to acknowledge my good friend and collaborator, Irina Ellison, who is not here today. She's currently at Mercy College, but also currently at another conference in Philadelphia. And I also need to thank the labs of both St. Francis and Mercy colleges. Both of our institutions have amazing teaching-focused collections related to biology. We have teaching slides and models, that help our students learn across biology. We don't have one bat skeleton, we have 20 bat skeletons. We don't have one microscope, we have 20 microscopes. And so Irina and I, in 2015, found ourselves in a position to both access these collections and also make them accessible. Plus, we thought it would be fun. We could think about our collections and the way that we teach science in a different way. So I'm going to walk you guys through our most recent event, and then I hope to see you guys at the next one. So what we're really aiming to do is to fuse artwork and lab work through events that invite anybody to have a scientific experience and re-encode it as art. And so in this picture, what I hope you guys can see or recognize these wave boats here, that we've filled up with high quality liquid watercolors. We have really nice supplies. We have beautiful models from our collections at our institutions. And we invite people to come and make work. Our events are always free. They're open to scientists. They're open to artists. They're open to anyone who is curious about science and art. And we try to theme them around concepts that might be interested to both artists and scientists. So for example, in thinking about cell division and mitosis, it's happening all the time. You know, in our bodies as we replace worn out cells, or if we heal a cut, or if we watch our children or our friends or our plants grow, we know that cells are dividing, yet in class we just learn the stages of mitosis. So we made an event called Control-C, Cell Copies in Growth, Regeneration, and Cancer, and it just happened last month at St. Francis, and it was partially supported by ASCB's Compass Grant, Compass Outreach Fund. And we had a full house. There were like 40 students coming in and out during the day, a mix of students, a mix of artists, a mix of friends, just coming through and not having to be engaged in a lesson, but actually looking at things and drawing what they saw. In addition to drawing pictures of cells in mitosis, we also did a very simple cell staining, looking at the morphology of transformed and regular cells. So everyone was able to do both the lab component and also a drawing component. So here's just a few shots from people who are not scientists who posted what they saw. We had people come in and make drawings from either the models or through the microscope. And it's just a very calm environment. We run it like a drawing class. And for those of you guys who love to draw, a regular drawing class consists of a couple of very short warm-up drawings, like 1-minute drawings, 2-minute drawings, because you know your first drawing is not going to be your best drawing. And then we move to longer drawings, like a 5-minute drawing or a 10-minute drawing. And at that point, everyone's in a pretty good groove. They know what it is they want to draw. And it is just a very calm afternoon of drawing pictures. This might be familiar to biology teachers in the room and probably most students. This is the mitosis in the onion root tip cell. And so here's some pictures that were drawn by bio undergrads who came into the lab. This is Aya Wadani. I'm sorry, I didn't put a credit on her here, but you can see that these drawings are really thoughtful. Students are taking the time to look at this material and engage with it. And I just want to echo what Greg said. Drawing used to be part of what it was like to be a scientist. Drawing helps us, whether or not we consider ourselves to be artists, but it can help us think. This is a classic drawing by Santiago Ramon y Cajal, and he was already introduced today by Greg, and he's famous for his drawings of the nervous system. These ones are drawings of cells in the retina. Cajal looked through the microscope. He would look at a bunch of sections, and then he would draw what he remembered. Now, in class, we show you a PowerPoint slide for 1 second, and we ask you to remember it and be able to answer questions on the test. I say, whatever you do in your studying, do something that helps you make that material yours. And so Irina and I were really excited once we started Art in the Lab that people were interested in it. People were coming to our events. People were enthusiastic about it. And we thought, oh, what would happen if we brought this into the classroom? Maybe we could help our first-year bio students learn stuff by drawing. So I'm going to tell you guys a funny short story. We've actually presented at MacCube in the past. So here's a story that we told at MacCube a few years back, and following some discussions this morning, I decided to put these slides into the talk. So we gave a little talk called Draw, Pair, Share, Activating Learning in First-Year Bio and Anatomy and Physiology Classes. And so what we did is that we had students and teachers come to, students in the class would just, I would give a short lecture, then students would get together into pairs, and half of them would explain to the other what was going on by drawing a picture. And then I would ask a question on the quiz, I would ask questions again on the midterm, and I would ask questions on the final and say, hey, did the drawers or the listeners understand it better? Okay, so the topic we chose to explore was resting membrane potential and action potential. And what I asked the students to do was just to draw the pumps and channels that are on the cell membrane. So as you guys know, there's all these pumps like the sodium-potassium pump that are always pushing sodium out of the cell and bringing potassium in. And then there's these channels that will destroy that carefully set up gradient by opening, and then everything rushes down as concentration gradient. And this is a very fundamental part of how neurons signal to each other. So I'm going to show you a couple student drawings. So you could have permeability, looking at charge differences across the membrane. Even if a student just copied the drawing from the text, I didn't care. I still counted it as a drawing because they were taking their time to learn it and really incorporate, or taking time to think about it. So short-term assessment, drawers outperform listeners on the quiz. And I was like, oh, that's awesome. Then I looked at the final, and I was like, oh, drawers outperform listeners on the final. Awesome. Then I did one extra experiment, and I was like, what about the people who didn't even come to class that day? Well, with the exception of one really disgusting story I told about the refractory period, the listeners just did the worst. Even people who didn't get to go to class did better than people who just laid back and listened. So what I have to tell you is just going to lecture is not enough. You have to figure out a way to make it yours. So I'm a printmaker and drawer by training. So in my art practice, I like to make drawings. I like to make prints from drawings. And I'm going to talk a little bit about printmaking because this idea of the copy, cell growth and proliferation, is also related to printmaking. So when we design the themes around the art in the lab, we're trying to think of something that could be interesting to artists. We're thinking about meme culture or appropriation or zines or anything like that. And so we had an artist come into lab who made this series of beautiful, beautiful etchings based on what she saw. But you can also make a print just from the surface of a leaf. And for those of you guys who have ever made a leaf print, you can just take a nail polish, a piece of packing tape, and put it on the back of a leaf, and then you can open it up and you can look directly at the surface of the leaf. So I would just say from microscope to sketch, whatever it is that you're doing, whether you're teaching yourself science or whether you're someone who just loves to draw, that you should make it work. Scientists and artists are both trying to get their heads around the world. You're trying to figure things out. And so I've always returned to drawing over and over again as a scientist if I'm trying to figure out how I'm perturbing a system, if I'm drawing a pathway, if I'm trying to make myself pay attention to the morphology of cells. So you could try it even if you don't consider yourself an artist. I hope to see you guys at upcoming Art in the Lab events. We're going to do one at the end of November, Mercy College, called Breathe. We'll do one in December called Body Rocks at SFC, and I'll talk to you guys a little bit about Body Rocks in a minute. January and February, there'll be an exhibition at St. Francis, so if you come to Art in the Lab, make a drawing, you can participate in the exhibition. And then we'll have another one at Mercy, and then we always do something Brain Awareness Week at St. Francis. So please don't be a stranger. This is where you can find us on Twitter and Instagram or on the web. Just that's where you can find out about where our next events are. So please come, bring a friend who has a serious eyebrow raised about science. Why not? So I mentioned Body Rocks, and so I'm just going to use this to quickly transition into the next project that I'm going to talk about. So when you guys are reading your textbooks, do you ever come across something where you're like, oh my God, that's so weird? So when I was an undergrad, I just loved cells crawling. I remember learning about cells moving, crawling through the basement membrane to the site of inflammation. I was like, that's so awesome. You know, the basement membrane is opening up, and then there's all this chemotaxis that happens, and you could just totally imagine it. And that didn't happen to me for a while because cells crawling got me so excited about cell motility that I decided to study axon guidance, which is how a neuron, newly born, has to send out a long axon, potentially over a long distance, to get to its synaptic target. But it happened to me again last year when I was like, holy crap, there's rocks in our bodies. I'm really interested in biomineralization. It's just so cool that you can have these cells that are secreting biomineral matrix. And so this was a print that I made this summer about this condition called crystallized lung that can happen in smokers or people who smoke methamphetamines, that they can get these crystalline inclusions in their lungs, including these cool things called asteroid bodies. And so this kind of fed into this larger body of work. This was a large-scale print that I made that was about astrocytes, which are the cells that help make bone. And this is maybe about a year or two ago. This was exhibited at St. Francis. And these are otoconia, which are the crystals that are in your inner ear that tell you if you are moving. So we're going to do an art in the lab called Body Rocks, and I hope that you will come. So scientists can use art or print technologies not only to help them learn stuff, but also to do science. So I'm going to show you guys some cool, interesting to me, examples of pieces of paper or drawings that were actually scientific instruments. So this is from 1512. This was a sundial type of thing that you could just like sort of, you could use it as a tool. And in contemporary science, we also use printing to print surfaces for culture cells to grow. One common example of this is a stripe assay, where you can use contact printing to put different patterns collagen or different patterns of chemical cues on your cell culture plate, and then you can see what neurons do, if they stick to the stripes or if they don't. Another example of a stripe assay. You can see that these neurons, their processes stick very closely to the laminin stripes, and they do not like the polylysine stripes. You can also do microcontact printing on human tissue or on cell cultures to try to prime cells for transplantation therapies. You can paint with cells, as you guys, sorry, you art science folks probably know about bacterial painting. Shout out to GenSpace, shout out to Katayun Chime, who does this at the New School. But it can also be a printed technology in that you can transfer these designs to fabric. Okay. So here's a little break. Seeing happens in the brain. I'm just going to remind you guys that you don't perceive with your eye, you perceive with your mind. So when you're looking at things, we've taken advantage of this as artists to bring you optical illusions, like how many triangles are here? 0. Are these lines parallel or do they slope? So these are, A and B are the same color. My students never believe me, but it's true. Intellectually, you guys know that this board is flat, but this work by Edna Andrade, who was working in the 60s in Philadelphia, even though you guys know this is a flat screen, it looks like it's moving or kind of bulging out. And so depth perception is pretty important when you're triangulating how far away something is. And so this was used as a type of camouflage for World War I. This type of camouflage is called dazzle camouflage. It's used now on basketball and soccer court, so it's a little more confusing to tell what direction your opponent is moving in or how fast they're moving. And this is what I studied in grad school. But I am not going to talk to you guys about GPCR signaling and retinal neurons. I am going to talk to you guys about seeing, because while I was doing these experiments in Penn, I started to think about how it is that we perceive things. I mean, vision is our primary sensory modality. And I started to get interested in how we see color. So quick reminder, you guys know that, we see the visible spectrum as electromagnetic radiation with a wavelength between like 400, 750 nanometers. I don't think you guys can read this, but these are spectra. This is spec analysis of random things in the environment, like classic art pigments like cadmium red, cadmium orange, or bright red, Winsor yellow, or cadmium yellow, or a cheese doodle, or a carrot, or an orange. So we perceive color in our environment in a different way. When we're making art as printmakers or as artists or just printing, then we have figured out RGB and CMYK, different ways that we can separate colors out and put them back together so our eye will reassemble them appropriately. In science, if you guys have ever taken a picture in the green channel and a picture in the red channel and merged them to see overlay in the yellow channel, it's a very, very similar phenotype. So I was thinking about this in 2013 when I made this sculpture, color spin sculpture in collaboration with my partner, Rob, who does large-scale print sculpture installation. And so we decided it would be fun to make a giant color spinning wheel and put it up at Socrates Sculpture Park, which is over in Long Island City. This is my nephew here, and this is a short video. I hope I'll be able to show you what happens when it spins. I made myself a matching skirt to match the.
Keep going. Don't stop. Keep going. Don't stop.
What's the point of going around?
Okay, you can stop and walk away from it.
So this was super fun, and it was a really fun break from graduate school. And I loved watching people come and interact with the sculpture, turn it, like little kids love the squeaking handles, parents and kids trying to turn the handle together, walking all around it and seeing how the colors were different. And yeah, it was really good. And it was a way where I could say, like, all right, this is the way that I was trying to think about how we see color. And then I could turn it into something that other people could experience. I'm going to use this now as a transition to move to talking about how artists are using scientific methods all the time to try to figure stuff out. So you guys now recognize this image, right? It's a photograph of that color spinner. Now, unless you guys are printmakers, you probably don't know that this is actually 4 different layers of colors. I had to make 4 different images, silkscreen them 4 different times, and then look at how the colors all layered up on each other. So I made these, were made using a method that I developed together with Rob at Frans Maasreel Centrum, which is a printmaking center in Belgium. And so we made a whole bunch of these different how-to videos, but the point is that we had to develop this methodology from scratch, and it was a lot like setting up a system in the lab. We had to make different calibration systems. Sorry. And we called our method fetching or fake etching because we really wanted to get some of the qualities, beautiful pigment qualities that you can see in etching and traditional print processes, but we wanted to do something that was less toxic, that was compatible with digital processes, and that anyone could do in their kitchen. So you guys probably recognize something like this. Maybe you've made something like this in your lab notebook. We ended up making a lot of tables like this where you would have, all right, here's the thing that we're varying. We're going to try all these different fine to coarse grit strengths of carborundum. We're going to try all these different varnishes. We're going to make notes about what these different conditions do. We're going to put red squares around the things that didn't work. We're going to put green squares around the things that didn't work. We're going to have notes over here. And so this was a really valuable way for us to figure out what was working and what wasn't as we developed this method, which you guys do not have to worry about at all. But basically what it was is that we use carborundum, which is the grit that is on sandpaper, to hold the ink so we could print it. So here's another one where we made these different charts like this, and we had all the different conditions that were laid out like that, and then we could triangulate how they were working. We could make little graphs, or we're like, these are the finest ones, and then this is how much ink we're adding. And it was incredibly useful. And I think what I like to say from this, or what I hope that you guys get from this, is that when you are trying to figure something out, science is an incredibly valuable way to think about stuff. And art is also an incredibly useful way to think about stuff because they're both methods for figuring things out. So I think I talked a lot faster than I planned to. I timed this for myself many times, and I had an hour, but I'm actually done. Don't be a stranger. Do you guys have questions? Question.
The bevel goes down and down every single time. Is there a way to salvage that so you don't have to strike out the plate?
Okay, this is an awesome question. So this was a question about etching and also related to what Greg showed today. So when you are making prints, you can either print a raised surface or you can print a engraved surface. So for Greg's paradigm that he showed today, he had an image that was exposed to light. The places where light could go through, that's going to be the positive, right? That's where that part gets baked on, right? Greg?
That's the high part of it.
So in our process, it's very similar. So it's actually not, we called it fake etching because the quality looks like aquatint. It looks like an etching, but it is actually a relief process because the ink is sticking to the sandpaper stuff, the carborundum. However, we have to make sure it sticks on the plate and keeps sticking on the plate. And so a good chunk of what I was showing you guys before was whether it was just going to fall off the plate. So all these red ones, the carborundum just fell off the plate. And so there was a matter of having the matrix there for the ink to stick through to, and also having that be adhered to the surface so we could keep our design there. So this is not actually an intaglio process to do, or like an engraving type of process. But when you, ways to preserve engravings, you can steel face a plate. And with this process, we actually found that you could get an intaglio line with a high enough bitmap. So this is extreme science science art nerdiness. But if you have a smooth varnish and a grit that's like 320, you actually do end up wiping into the grooves, and you have an intaglio like mark, but you'd probably only get like 10 or 20 impressions out of it. Greg.
Do you have examples of when you were in grad school, you kind of were drawing things out or using any kind of artistic technique to understand your
I find drawing to be extremely helpful for science in thinking about cell signaling pathways or what happens in mutant situations, like loss of function situations. So when I was studying G protein coupled receptors, like when I was looking closely at cell signaling, I would draw out all the different pathways, and then I would just imagine, or then I would literally erase parts of them or add something to them or put something over them. And then I could imagine better or literally picture what would happen better if I could change something about that pathway, because you have to really visualize it. I think about that when I'm thinking about receptor signaling. I think about that when I think about regulation of receptors at the surface of the cell membrane. I use drawing to think about mutant phenotypes, because you guys know genetics has the double negatives, because they name the mutant the thing that the phenotype was, which is usually the opposite of what the actual molecule does. And it can get confusing when you're on an exam question. So I think one thing really helped me is to say, like, oh, they call this one little, but that's actually because it's promoting bigness, whatever. So, this because the names are the opposite of the function, I feel like drawing those situations has been very helpful for me, and maybe for other students too. you mean that incorporating art into the learning process has changed the student's career path?
I guess it's a two-part question. One, do you think you affected some career path of some of the students? And the second part, do you think you affected how students approach late learning other portions?
Oh, I don't know. I hope so. I mean, I hope that I have not done that assessment. I haven't done that experiment to say, like, I had some students where I never used drawing and I had some classes where I did, and what happened to those students long term, or to test the same concepts in different classes. So, like, let's just say we do action potential freshman year, and then in cell biology, and then in neurobio, and I could see, like, when you learned it, and then how you perform when you learned it in freshman year, whether you were a drawer or a listener, and then when you take neuro when you're a junior, if you take a pre-assessment, see how well you remember it. I haven't tried to do that, but I think I might try that. Yeah, there's we did so in that experiment that I showed you with the drawers, the listeners, and the people who weren't at the lesson. So we've repeated that, and this time instead of letting drawers and listeners self-select, we randomize it, and the drawers still do better. So I think part of it is just about giving yourself the time to figure out in a way that's meaningful for you. Question.
I know why. I just because I feel like when I was in school, I couldn't figure out how it worked. So I could just make sense of it. Like first this or that is like I mean I don't I think when you're drawing and the student won't have to get sufficient time in the classroom.
It does take a long time to draw, and for those of you guys who take intro majors bio or teach it, you know there's not a lot of time. We have to move really fast. And so I can say that it does take some time. It's a 10 to 15 minute lecture, then it's 10 to 15 minutes to do, I mean, really, it's 10 minutes, but in reality, it's 15 minutes. And then you give a short quiz, so that takes a little bit of time. But when you look at the drawings afterwards, you can see really quickly what it is people are not getting. You can say, oh my goodness, this person thinks that channels are always driving things out of the cell. Or, oh, I see that a few students have done this thing, and so I know I can make that more clear. And I think that's super valuable because as a professor, you want to know that your students are not understanding something before the test, because then you can fix it. And so in some ways it does take time, but in other ways it is a really fast way to get feedback. And Irina and I certainly saw that, but we weren't the first people to see that it's a very fast way to get feedback about student misconceptions. So you can draw something one way and be like, oh, okay, I see what you're seeing there, and actually I can fix it.
In your share pair groups, did you ever try doing collaborative drawing where a student starts the drawing and then the other adds to it, or takes something away, or adding to it would probably be the most?
I would love to try that. That would be a great thing to try. We haven't done that. Irina has also pulled this into her anatomy teaching stuff to try her course material in anatomy physiology. And I'm sorry I wasn't able to present her work today. Great suggestion to try to have students work together to make a drawing, build a drawing collaboratively. So one person would be adding things to something another student did, and we haven't tried that, but I think it would be amazing to try it.
So there's different kinds of learners. There's the visual learners, the
auditory learners. So my question is, have you compared the retention of information for students that took the course with drawing incorporated versus students that took the traditional course without the drawing being incorporated? And to see that class has better retention of the material at the end of the semester compared to the traditional class.
So it's a great question. In some ways, I can't really control for it because now I've put it into, I would have to step back and stop including it because at the time when I started to include drawing, I am still learning how to be a teacher now, but I feel like it wouldn't be quite right to compare my first year teaching results to my 6th year teaching results. But if I had 2 sections of the same course and one had drawing and one didn't, then I would be able to do that comparison. It might be really interesting to look at that. Yeah. Questions? The question, a really important question, the question is about money. What are the agencies that will support this kind of project? And I think this is something that we all who work in art and science are thinking about. First of all, Greg mentioned today that the National Science Foundation does support art and science projects, particularly when they're related to outreach and teaching. ASCB Compass, for those of you guys who are members of professional organizations, I know that SFN is recently doing more and more with art and science. That's the Society for Neuroscience. I believe Society for Biochemistry and Molecular Biology, B&B, they do stuff. So I would say your professional organizations probably have some grant money reserved for science outreach, and that's definitely a place where you can ask for funding for these types of projects. The Simons Foundation has been really instrumental in looking at this. And then there's also smaller or more arts-focused organizations that, particularly those that are looking at socially engaged art. To do a project like Art in the Lab, where you're involving a lot of people and inviting them into the lab. If you have your own personal art project that you would like to be funded and is related to science, then I think that's a different story. But as far as outreach goes, I think that you can get a lot of traction through professional societies, NSF, Simons Foundation. To do collaborations between the art and the biology departments in the classroom? So this is a great question, too. So the first thing is that St. Francis College has amazing labs, but it doesn't have a studio art program. But there are other colleges, for example, Pratt doesn't have labs. They have amazing studio facilities. There are architecture students and studio art students who are interested in biomaterials or interested in biodesign. And so those are opportunities, I think, always. And our college now has a new Department of Interdisciplinary Studies, and I'm really happy that I'm a member of that department, to think about how we can go across departmental boundaries, because we want our students and we want to ask questions, big questions that you can't just maybe use one discipline to solve, because complex problems are more than one discipline big. Okay, I'm gonna appropriate Greg and say 3, 2, 1. Thank you.